Over the years that we have been critiquing germ theory, virology, and the concept of microbial pathogens, our work has inevitably extended into the animal world. On this front, we have fielded increasing numbers of questions relating to the health of both livestock and household pets. After seeing the fatal flaws of allopathic medicine, one comes to realise that veterinary medicine is also dangerously steeped in the same practices. However, one of the most common questions we get is, but what about parvo? It is a fair diagnosis, as it comes with high fatality rates, and the establishment claims that only vaccines are effective in preventing this alleged viral disease. Let's have a look at some of the problems with this narrative and the fascinating story of so-called canine parvovirus.
Welcome back. Emergency veterinarians across the state are seeing a startling wave of parvo cases in family dogs.
Yeah, parvo is not good. It's a nasty disease. Left untreated, the mortality rate is very high, about 90%. That percentage drops below 50% depending on the type of treatment. And even surviving dogs are vulnerable to lifelong health issues once they get it. Joe Dana tells us why vets believe this outbreak is happening.
Veterinarians tell us they're not just seeing an outbreak in parvo in puppies, but in adult dogs as well.
Look, how's she been doing?
Doing good.
It's booster day for Rage. Rage's owner takes no chances.
She was sick when she was a puppy and spent 10 days in intensive care.
Not all dog owners are so vigilant with shots. It's that complacency that veterinarian Thomas Satkis says may be one reason parvo cases are up.
We know it's bad because we go through a lot of parvo tests. We've been seeing a lot of puppies that we're diagnosing with parvo, and we're even seeing older dogs being diagnosed with parvo, which isn't typical.
But there's something else going on. The Peoria Animal Hospital and 3 24/7 clinics around the valley tell 12 News they're seeing an unseasonal surge in parvo cases, with multiple dogs in intensive care at once. Clinics and social media groups are spreading the word online. Some vets say record-breaking autumn rains across the state may have spread parvo in waterways and where dogs play.
Parvo is a virus that's spread from the poop of other dogs, and it can live in the soil for up to 7 years. We don't see dog poop from 7 years ago. It's just dirt, and that's how dogs end up, unfortunately, getting parvo.
As breeders cater to potential buyers during the holidays, Satkis says be sure puppies begin the first round of parvo vaccines at 6 to 8 weeks. Boosters are recommended for adult dogs every 1 to 3 years.
It's almost impossible to over-vaccinate a pet. So if there's any question, start the series again.
What is canine parvovirus? This is where the first problems with the alleged specific diagnosis begin. Wikipedia's canine parvovirus entry states, the signs may include lethargy, vomiting, fever, and diarrhoea, usually bloody. Generally, the first sign of CPV is lethargy. Diarrhoea and vomiting result in dehydration that upsets the electrolyte balance, and this may affect the dog critically. Secondary infections occur as a result of the weakened immune system. We can ignore the scientifically unfalsifiable concept of a, quote, weakened immune system in this description. If you are unsure why this is so, then please watch my video, Is Immunity Real? And, quote, secondary infections can also be ignored in the same vein. As we have discussed many times, the correct use of the word infection is its original meaning. That is, the tissue is stained or ruined, not being attacked by microbes, although they may certainly increase in number. Once those points are appreciated, the key factors in the dog's illness boil down to the prolonged severe diarrhoea and vomiting. This cannot be considered a distinct disease, of course, as both signs are simply eliminatory processes. If they become prolonged, then naturally further compromises to the dog's condition are inevitable. While the Wikipedia entry goes on to state, dogs have a distinctive odour in the later stages of the infection. This is a result of the ongoing reaction rather than some special property of the alleged virus. Severe diarrhoea and vomiting typically results from the ingestion of a toxic substance in the animal's food or water. Additional toxins to consider are veterinary drugs and accidental exposure to man-made chemicals. Keep in mind that different animals have variable sensitivities to specific compounds. For example, eating dark chocolate is not dangerous for us, but potentially fatal for a dog. Also consistent with the toxicology theme, younger dogs diagnosed with parvo do worse than older ones. A similar pattern was seen with human polio cases last century. Along these lines, we see the suspicious historical pattern when the alleged viral parvo epidemic suddenly appeared on the scene. Again, this is reminiscent of polio last century. If you are unaware of the polio story, then you can find out about it through our book Virus Mania or through F. William Engdahl's Toxicology versus Virology, Rockefeller Institute, and the Criminal Polio Fraud. There is also the original toxicology work of Jim West, including my 2022 interview with him. I'll put links in the description for all of these important resources. Coming back to the parvo narrative, Wikipedia reports that Parvovirus CPV2 is a relatively new disease that appeared in the late 1970s. It was first recognised in 1978 and spread worldwide in 1 to 2 years. Well, we know from the COVID-19 fraud that no virus is required to spread worldwide. All that's required is a fear campaign and the rollout of a new test, which we'll get to soon. However, equally so, it may be that a new wave of sickness did appear in some clusters of dogs in the 1970s, but an imagined virus was not the cause. In December 1978, Status Report Canine Viral Enteritis was published in the Journal of the American Veterinary Medical Association. The authors reported that episodes of widespread outbreaks of contagious vomiting and diarrhoea, sometimes hemorrhagic, first were reported in February 1978 to the Cornell Research Laboratory for Diseases of Dogs. The key word here is contagious, and there is a single citation. The problem is that this citation is simply a laboratory report rather than a formal study. The author Carmichael did go on to publish a more detailed paper in the Cornell Veterinarian in 1979. But wait for it! The first sentence of this paper is, widespread outbreaks in dogs of contagious vomiting and diarrhoea, sometimes hemorrhagic, first were reported to us in February 1978. Again, there is a single citation, and it is Carmichael's same laboratory report. This self-referential process exposes the lack of scientific rigour in the foundations of the alleged parvovirus and the wider pseudoscience of virology as a field. To be clear, none of these reports established that there was a contagious microbe as the cause of the dog's illnesses. They simply make comments such as, particularly severe outbreaks occurred in veterinary hospitals, boarding kennels, and commercial or research kennels. In the 1979 paper, the authors performed electron microscopy on dog feces and claimed to identify various viruses. On some occasions, they reported, quote, both the coronavirus and the parvo-like virus were identified in the same stool sample. However, these were simply point and declare exercises where the nature of the particles had not been established in a scientific experiment. If you want to know more about this shonky aspect of virology, then you can watch my video on electron microscopy. In this case, the authors believe they can distinguish between the two imagined viruses, being canine coronavirus and canine parvo-like virus, although do admit that it is difficult to make a clinical differentiation in most instances. Essentially, it is a case of them being gastrointestinal disturbances, with parvo being at the more severe end of the spectrum. With regard to the electron microscopy, in none of the examples had the imaged particles been shown to be A, the cause of disease, or B, viral in nature. The authors state, the coronavirus is highly contagious, and it spreads rapidly through kennels of susceptible dogs. It should be noted that the word susceptible pushes the contagious claim towards an unfalsifiable paradigm. There is no detail about what they did or how the so-called contagion experiments were controlled, simply a statement that experimental CCV infection was, without exception, mild. Similarly, with alleged parvovirus, the only information we are given is leukopenia is often accompanied by fever. Both signs were prominent in experimentally infected laboratory beagles. This is followed by a later admission that the experimental disease has not been adequately studied at this writing. At this point, we might suspect that they were forcing the dogs to ingest feces from a sick dog. If they are claiming that a virus was the cause, then a control experiment would need to be done, where the same feces was fed to dogs, but this time with the alleged viral particles removed. As far as we know, this has never been done in the history of virology. But it gets worse than that, because we can finally expose the details of the experiments in another 1979 publication that is the linchpin for the entire parvovirus story. The paper appeared in the Veterinary Record and is titled Isolation and Immunization Studies of a Canine Parvo-like Virus from Dogs with Hemorrhagic Enteritis. Interestingly, it is a paper that is not readily available to the public. So let's shine some light on it and address the headline's claim that they isolated a virus. Under the quote, virus isolation section, they reported that the feces from dogs with hemorrhagic diarrhoea was passed through a filter with a pore size of 200 nanometers. This crude extract was then added to feline kidney, canine kidney, and a mink lung cell line. As we all know, those are the cell lines that we expect to handle dog poo. Next up were the equally inappropriate feline lung cells, veromonkey kidney cells, raccoon salivary gland cells, and bovine fetal spleen cells, all poisoned by the addition of dog poo, with the claim that the mixture qualified as, quote, isolation of a virus, and a mostly dog-specific virus at that. At no point did they show direct evidence for a virus, but then they make the remarkable claim that the presence of viral antigen was demonstrated by fluorescent antibody tests, or when hemagglutinin was evident. There is no way to conclude that they had a, quote, viral antigen and an associated antibody when no virus had been physically isolated to begin with. But as my husband Mark said in his keynote speech at the Wise Traditions Conference in Salt Lake City last year, assumption is the mother of all virology. The notion that hemagglutination demonstrates the presence of a virus is equally dubious. This, quote, test dates back to the 1940s, when it was proposed for the detection of alleged influenza viruses. It was claimed that virus glycoproteins cause the clumping of some types of red blood cells. While it may be true that red blood cells clump together in the presence of certain glycoproteins, there is no evidence that the glycoproteins come from viruses. In the case of alleged parvovirus, the cell culture did not cause red blood cell agglutination in species such as mouse, rat, chicken, guinea pig, hamster, human, and dog, but did cause clumping in pig red blood cells. Therefore, virus.
That'll do.
Don't be fooled by the later statement that known negative and positive control sera were included in each test series. They have not provided evidence for a virus, so how can they know in advance whether the samples are positive or negative for a viral illness? Then we get to the alleged contagion experiments. Did they feed the test dogs some fecal material, you may ask? No, they injected fecal extracts straight into the veins and noses of 4 beagles. And what was the reaction to this unnatural exposure route? They reported that only 1 dog given an intravenous dose developed a fever. Then they used a fecal cell culture mixture to perform intravenous injections into a further 8 young beagles and reported some elevated body temperatures and changes in white cell counts. However, it was candidly admitted that no dog developed anorexia, diarrhoea, or other severe signs of illness during a 2-week observation period. So not only did they use a preposterous exposure route that doesn't replicate what happens in a natural setting, but the dogs did not even develop the typical illness, that is, with gastrointestinal signs. Additionally, there was no scientific method in sight because there were no control experiments. There should have been a comparable group of dogs that were injected with a fecal cell culture said not to have the virus. The experimenters should also have been blinded to what they were administering to the dogs. However, please don't get me wrong. I'm not suggesting that this methodology was either humane or appropriate to demonstrate contagion in the first place. The subsequent, quote, immunization studies were equally pseudoscientific. These were based on antibody titers, which means concentrations at which the assay was set rather than presence or absence. As these tables reveal, if something was injected into a dog, whether it was called a vaccine or a virus, then the assays typically became more reactive. Once again, you can watch Is Immunity Real to appreciate the fallacies of the antibody concept and the related laboratory assays. In any case, we haven't taken our eye off the ball because these subsequent studies were invalid as they did not show the existence of a virus in the first place. However, it was publications such as this that led to the worldwide rollout of the parvo vaccine. And it was Leland Carmichael at the Baker Institute for Animal Health at Cornell University who was pivotal in the development of this veterinary vaccine and others. Indeed, the Institute's 1982 annual report stated that virtually all the vaccines now used to protect dogs against distemper, hepatitis, parvovirus, and other diseases caused by viruses and bacteria grew out of research undertaken in the Institute's Colgate Tissue Culture Laboratory. Leland Carmichael headed one of their laboratories, which explains his prime position in all of the parvovirus publications. During this time, we also note a familiar name on the Institute's Advisory Council, being William M. Rockefeller, at that time also a partner at the White Shoe law firm, Shearman and Sterling. The list of supporting organisations included the Geraldine Rockefeller Dodge Foundation, and the connections did not stop there. The Baker Institute was named after Dr. James Andrew Baker, who came to Cornell in 1947 from the Rockefeller Institute. It was there that he was said to be heavily influenced by Peyton Rue, and those of you that have read A Farewell to Virology will appreciate the significance of this. In all, a world of Rockefeller-sponsored activities geared towards promoting the virus model and the derived pharmaceutical products. Dr. Baker died suddenly at the age of 64. But no, not what you might be thinking. It was 1975 and the smokes probably hadn't helped. His protege, Leland Carmichael, died at the age of 90 in July 2020 during COVID mania. On his death, one of his former colleagues remembered a letter which stated that Carmichael had done more for the health of animals than anyone since Pasteur. It seems that this was meant to be a compliment, but in reality, Louis Pasteur has been exposed as a fraud who needlessly killed an obscene number of animals while contributing to the fraudulent practice of vaccination. To give some credit to Carmichael, in 2005 he wrote that with regard to the history of parvovirus, the news media had a field day reporting outbreaks, reports that often created extreme apprehension because of sensational coverage and fabricated myths. However, he still believed that he had helped discover a virus, and the subsequent vaccines were saving thousands of dogs. On that account, the global dog vaccine market was estimated at $2 billion in 2024 and is projected to reach US $2.88 billion by 2030. Parvo vaccines are a huge part of this market, and according to the Cornell College of Veterinary Medicine, these shots are given every 3 to 4 weeks from the time a puppy is 6 weeks old until he is at least 16 weeks of age. A booster vaccination is recommended 1 year later and then at 1 to 3 year intervals thereafter. It's a superb business model that is embraced by vets who then allow their clients to pay for it through the parvo fear narrative promulgated by the corporate media.
Okay, so anything else we should know about parvo?
The biggest things are vaccinate, vaccinate, vaccinate. Yes.
The Cornell article also states that in order to protect a new dog, you should not bleach your lawn, but rain or watering can dilute the concentration of the virus over time. As we move further into clown world, the College of Veterinary Medicine homepage is evidently advertising its involvement in testing weight loss pharmaceuticals on cats. There could be a joke about fat cats here, but the twisted depths they have descended to under the guise of, quote, animal health is no joking matter. Animals get sick for similar reasons that we do. This includes poisoning from the environment and food sources, overeating and obesity, lack of exercise, malnourishment, and chronic stress. Additionally, most dogs are natural scavengers and eat indiscriminately, a behaviour that puts them at further risk in the modern human world. Parvovirus is yet another cover story for why animals really get sick. As I mentioned earlier, clusters of dogs developing gastrointestinal signs with higher fatality rates in the younger pups points towards a toxic exposure. However, such causes do not provide huge revenue streams for the industry and would potentially expose embarrassing problems somewhere else. Unfortunately, many boarding kennels, doggy daycare organisations, and even group training activities require proof of parvovirus vaccination. Like the COVID vax passes, I would encourage everyone to think about the real consequences of participating in these rituals. If you have more questions about this material, then please check out all of the free resources at drsambailey.com. In addition to your health matters, we also cover how we look after our pets and livestock in our fortnightly Q&A sessions. If you enjoyed this video, please visit supportdrsam.com.